hepatitis A and E virus. In this session, we will talk about these two viruses. Both are single-stranded RNA viruses and very much common in Pakistan. In this session, we will talk about the occupational risk of these viruses to lab personnel. Now let's talk about hepatitis A virus first. It's a single-stranded RNA virus and belongs to a genus known as hepatovirus, while the family to which hepatitis A virus belongs is Picornaviridae. This is the structure of hepatitis A virus. Here you can appreciate the head portion of hepatitis A virus as it is enclosed by a capsid containing single-stranded RNA, and here you can appreciate the viral polypeptides as well. There are 4 types of viral polypeptides: VP1, VP2, VP3 and VP4. These viral polypeptides helps in the pathogenicity of these viruses. Now let's talk about the second virus, that is hepatitis E virus. Just like hepatitis A virus, hepatitis E virus is also a single-stranded RNA, but it belongs to genus Hippivirus, while it is not assigned to any family because it has a floating genus. That's why it is not assigned to any family. Otherwise, hepatitis A virus belongs to Picornaviridae, but hepatitis E virus does not belong to any family because of its floating genus characteristic. Now, hepatitis A virus and hepatitis E virus is not a major occupational risk among laboratory personnel. However, hepatitis A virus is documented in various reports when the laboratory personnel is handling any infected animal. For example, if the infection of hepatitis A virus occur in chimpanzees or in other non-human primates, then it can be transferred from the animals into the human beings. Now, hepatitis E virus has a very low risk of occupation among lab workers as compared to hepatitis A virus, except during pregnancy. If hepatitis E virus infection occur, then it can prove to be very severe and even fatal. Now keep in mind that hepatitis A virus causes both foodborne and waterborne diseases, while hepatitis E virus is only a waterborne disease, and it is acute enterically transmitted disease that causes hepatitis. So, if the water or if the food is contaminated with hepatitis A virus, then it can lead to hepatitis A. In the same way, if the water is contaminated with hepatitis E virus, then it leads to hepatitis E. And both of these hepatitis are very much common in Pakistan. Now, this is occupational risk. If you talk about the occupational risk of these two viruses, then it causes primary hazard to lab personnel, because whenever the sample comes to the laboratories, the workers has to deal with these samples, and the samples come in the form of stools, in the form of feces suspension, and even in the form of other contaminated material. Even blood serum can also be used for the serological diagnosis of hepatitis A and E virus. Now, if the lab worker is suffering from any injury or if there is any wound, then care should be taken, because if the worker is handling any contaminated material, then these viruses can easily penetrate through the wound punctures or through the injuries. इसके अलावा अभी तक एरोसोल एक्सपोजर के थ्रू हेपेटाइटिस ए वायरस या हेपेटाइटिस ई इन्फेक्शन का अभी तक कोई रिपोर्ट सामने नहीं आई. Now, in order to deal with these two viruses, we prefer BSL-2 practices procedures. इसके अलावा हेपेटाइटिस ए के अगेंस्ट इनएक्टिवेटेड वैक्सीन्स अवेलेबल हैं, but currently no vaccines are available against hepatitis E virus.